Alright, hi guys, welcome back to my channel. My name is Alexus and I am a third-year PhD student studying Biological Oceanography at the University of Hawaii at Mānoa. Okay, so to put it very simply for you guys, because I'm not sure I've mentioned this in a while, I am studying Zooplankton Ecology in the Eastern Tropical North Pacific Ocean, as well as impacts of deep sea mining in this area. Okay, so it is December 7th and I wanted to make a quick video about my progress on my PhD this semester. Now that the first semester of my third year is coming to an end. This means that I'm at exactly the halfway point. If my PhD is going to take 5 years, who really knows, but I think I'm at the halfway point, which is pretty bizarre. But yeah, I just wanted to give you guys a quick update about what I've been working on and how my PhD is going. Okay, so just to give you guys a little bit of context on where I'm at in my program, I was actually taking 0 classes or courses this semester, with the exception of an advanced stats in R class that I am making up an incomplete for. So I was taking this course while I was on a cruise, and because of just the amount of work that I was doing on the cruise and applying for the GRFP, there wasn't enough time to finish this course. So I'm still making it up and I have about 5 homework assignments to complete by the end of the semester. But other than that, I am pretty much done with my coursework, which is a really great feeling. I had 0 cruises this semester and 0 fieldwork this year, which means that most of which I've been doing is pretty much just lab work from the first 2 cruises in 2021. These were both 5-week cruises where I went out and collected a bunch of zooplankton samples using nets. So I'm kind of still going through all of those samples from the cruises. Okay, so before getting into what exactly I've been doing in the lab this semester, I wanted to give you a brief just summary of what my research is exactly, so you have a decent understanding of what the different goals are for my projects. So what myself and my lab are working on right now is collecting baseline data on the zooplankton community that is found in the eastern Clarion-Clipperton zone, which is an area that will be impacted by deep sea mining of polymetallic nodules. So before any of this mining begins, we want to have a really good understanding of what's there in the different layers of the water column from the sea surface to the sea floor. We want to be able to describe those species, how they contribute to the community structure and food web, and what this means if deep sea mining happens. So in order to describe this zooplankton community, we surveyed zooplankton using nets, and I'll be analyzing them using a combination of molecular and morphological approaches. Okay, so the main project that I've been working on in the lab this semester, which I actually began in October 2021, so over a year ago, is the DNA metabarcoding project from the first cruise. So I finished DNA extractions from these zooplankton samples last semester around March and April. So I basically spent the whole summer trying to amplify 2 regions of the genome that I'm specifically interested in looking at. So this is 18S and the COI gene. And I'm doing this to determine which organisms, so which species or certain taxa are making up the zooplankton community at these different depth layers in this specific area of the ocean. So these data are going to give me a really good idea of who is here and who is going to be impacted the most by deep sea mining. Okay, so from summer basically up until now, I've been working on amplifying those regions for 218 samples. So doing lots of PCR pretty much every day in the lab 9 to 5. And I've had some pretty problematic samples that were just taking forever to amplify. I had to do it so many different ways in order for it to work. And then after PCR, I've been doing DNA purification, so just bead cleaning to clean up the PCR products and get them ready for sequencing. So before Thanksgiving, I submitted my first half of samples for sequencing and am just getting ready to submit the second half this Friday, so in a few days, which is very, very exciting. The first half that I already submitted has already been returned to me, which means that I have this data in hand, and the next steps are to figure out exactly how to analyze these data, because I've never worked with sequencing data before, so I have close to zero idea on what to do once we get back all of these reads. So yeah, that's the next steps for that DNA metabarcoding project, is just submit that second plate this Friday and then start working on the analysis. That's not going to get done by the end of this year, unfortunately, but I'll have plenty of time to learn how to analyze that data, do the analysis, interpret it early next semester. Yeah, I'll have so much time, so that's the next big thing. The second project that has pretty much been running in the background without me, big thanks to my lab undergrad, who's been working on zooplankton biomass measurements for the second cruise. So these are just bulk wet and dry weights for zooplankton that were collected in the nets. It is not taxon specific, unfortunately, so we just know roughly how much zooplankton were captured at each depth layer, but we don't know what makes up these different communities. However, these data are really, really useful because it allows us to look at zooplankton deal vertical migration behaviors. And we're working on the second cruise now so that we can make comparisons to the first cruise and determine if there are any seasonal differences, since the first cruise was done in the spring and the second cruise was in the fall. So my undergrad's been the one in the lab every day collecting these measurements. I've kind of just been overlooking this project while I've been doing all of my PCR. But she finished it. She collected all of the measurements for me. So now I'm working on the data analysis portion of this project. So I just spent most of the day yesterday and today working on biomass plots for the second cruise. So the next steps for me within the near future are to finish up a lot of this data analysis specifically for the biomass project, because we have a big grant deadline at the end of this year, which means we're going to do a really big data dump, because I kind of just got back all of that sequencing data, and my lab mate who's already done a similar analysis is going to do a very, very quick first pass data dump for me, luckily. Because it would take me a really, really long time to learn it. Far too long given our deadline. So I have lots and lots of plotting to do this week as well as the next up until that deadline. And I also need to magically finish my incomplete R course. I still have 5 homework assignments to do. It might not sound like a lot, but those assignments take anywhere from like 4 to 6 hours for me to do. So I don't even know if that's feasible anymore. We'll see. My fingers and toes are crossed, and please cross your fingers and toes for me. But I'll update you on whether or not this actually gets done. Originally, I was supposed to be taking my comprehensive exams and defending my prospectus at the end of this year or this semester. However, given all of the lab work that I've been doing, I just really have not had any time to do thinking towards my dissertation, which means that I'm putting my comprehensive exams and my prospectus defense off until next semester, probably around March, so that I have enough time to study, get all of my ideas together, and do a ton of reading. I have so much reading to catch up on. I'm going to be taking a huge step back from lab work after the holidays in order to work on all of the thinking and reading that I need to do for my dissertation planning and my comps. and prospectus, but I'll also be mentoring and overseeing my undergrad who's going to be working on zooplankton DNA metabarcoding from the second cruise. So we'll also be able to make some comparisons between the first and second cruise for those data. So really just a lot of things have been getting done in the lab. Everything's kind of been wrapping up all around the same time, which is nice. But this past semester, I have been doing way too much lab work and not enough of everything else. So I've really fallen behind and just completely fallen off track when it comes to the reading. I haven't been doing a lot of data analysis because I've been focusing on generating these data in the lab. So yeah, it has been a lot, and it's been a rough semester. Mentally, I'm in an okay place now. Can't really say that I was a few weeks ago. I was getting sick of PCR, sick of molecular work, just totally sick and could care less about my project. Now that everything's kind of finishing up, I'm doing more data analysis in R, a little bit more computer work. My life feels a little bit more balanced, and I'm not going as crazy as I was a few weeks ago or a few months ago. But yeah, I think that's it for this video. Hopefully I haven't rambled for too long. If you guys enjoyed this video, I'd really appreciate it if you give it a big like because it really helps me. Subscribe, and I'll see you guys in the next one. While you're waiting for me to post more videos, why not check out some of my PhD vlogs? So I try to film weekly vlogs, a day in my life, work-life balance, I don't know. Go check it out.